I'm Jack Anthony. At 6:30 this morning, I get a phone call from Maury Ruffin saying, "Get your ass up to the city. We need you to speak." Why would he call me, other than I've known him and Carola and Sarah for years? I actually was at a cell therapy company, Applied Immune Sciences, with Tom O'Karma in the late 80s, which was a predecessor to Dendreon, which was then called ACT when it emerged. And when I was at Baxter for 17 years, I worked with Steve Rosenberg at the NIH and with Kurt Sivan out of Massachusetts on stem cells and lac cells. And since then, I've done 9 companies. I've sold 4, tanked 1, and I'm on my 9th one now. And this may well be one of the most interesting technologies. It'll apply to everybody in regenerative medicine at the end of the day. And so it is a little different, and I kid you not when I say no one else is doing what we are doing. So we'll go from there, and let's do it. What we've got, maybe you want to bring the microphone. I'm a pacer when I talk. Got the microphone right there. Beautiful. Okay, thank you. Hello. Hello, hello. Okay, we are a biomaterials company, biomaterials that are highly biologically active. And we do this in a very highly proprietary manner. Think about, let's use the one collagen, for instance. Collagen is used in thousands of products. It's really kind of a carrier. You might put some thrombin on a pad of collagen and it becomes a way to stop bleeding or other things. Collagen then is used as sort of a carrier for a message. The way we have reinvented collagen is the collagen actually tells cells what to do. And that's what happens in your body when you think about it. You have over 21 different kinds of collagens in your body. It is 25% of all the protein you have in your body. And the collagens are telling cells what they should be doing. Okay? And so we have an amazing effect on telling various cells, proliferate, stop proliferating, go left, go right, whatever. That's what we're all about. And I'll walk you through this. Okay, we are starting with collagen because it is ubiquitous and there's a lot of usage of collagen in the world out there. Collagen used to be considered, well, it's the stuff that holds your body together. But it's much more than that. So you can see that is ubiquitous in various organ and tissue systems through the body. This is material that we have made in our process. And it imitates, if you will, exactly the various tissues in your body. The science came from Silicon Valley, where our Russian physicist, mathematician, was one of the early inventors of flat-screen LCDs. And he sold the company to a Japanese group. He moved to the Stanford Medical Center, where he was doing some work as a professor. And it occurred to him, whoa, collagen is a liquid crystal material. I wonder whether I can handle that the same way I used materials when I was making LCD displays down in Silicon Valley. And what he found was, and I won't go to all the details, but when you process collagen, I don't care whether it's recombinant human, cow, bovine, chicken, what it is. It's your choice. The process that you use in LCD production changes ordinary boring collagen into tissues. And we can add other materials, we can recombine materials, we can, in fact, build tissue at Fiberline. And we do this, with this process, we can change the mechanical nature of the materials, we can change the physical nature, and we can change the biological effect of these various materials. We're working with some strategic partners now. One wanted a dural covering post-cranial surgery. They wanted it transparent. The only product on the market today from Integra is very dense and opaque. And we delivered in 6 weeks 6 different samples of materials of various strengths, but all of them transparent, all of them conformable to the surface of the brain, and all of them containing very little collagen, so we're very economical. This is the stuff you buy today. Years ago, you used to buy a collagen membrane in a casting methodology. There's electrospun collagen out there and lyophilized collagen, okay? This, however, this is what native collagen looks like. These materials look nothing like what's in your body. And therefore, just using collagen out of a jar has no effect on cell communication. It's not helpful in cell culture, and it's damn well not helpful in tissue engineering. We make materials, as I showed you before, that can emulate any tissue you want just by dialing in the parameters. Okay, so our materials, by letting the collagen self-assemble back into what it was through a process, eliminate all the bad things that happen when you electric spin collagen into a pad. Okay, there's a lot more behind this, but I'm giving you the helicopter view. We can make it in various formats for you. If you'd like it in sheets, be happy to do it in sheets. We can turn these sheets into various woven forms. We can make these threads out of it, and we can make these threads that will really hold a lot of weight on them. This is a cross-section of the thread, and this will come back in the presentation. There are actually orifices and tunnels in the thread, and we can make tubes. We've got experiments going at Stanford right now with some neurologists where they're inserting broken nerve ends each end of a tube, and because of the alignment of the collagen, it tells the nerve, grow together again, so it knows exactly where to go. Now, this is one of my favorites. When I was in the collagen business back in the late 90s with FibraGen, not to be confused with FibraLine, that was back during a mad cow era. We thought we had a license to print money. We were making recombinant collagen. Away went mad cow. It was never found in any medical devices anyway. So I knew enough about collagen to be dangerous. This is an experiment with elastomer tubules, these little short, stubby tubules on a plate. Some of them are very short, and some of them are very tall, relatively speaking. And what this will demonstrate is by taking mesenchymal stem cells that are really kind of neutral and dropping them on various heights and flexible elastomer tubules, it tells the cell what to do. In this case, the rigid one said go osteogenic, and the very flexible one said go adipogenic. And so there is communication between stiffness, if you will, and what a cell will do. And that's exactly what we can build in, one of the parameters to the collagens that we turn out. This is a complicated, messy slide. I'm sorry, I probably should have edited it out this morning, but I was driving up here. The take-home message here is this is work out of Professor Desai at UCSF, where she took corneal cells. She challenged them on collagen, both the stuff you buy from VMR, whomever, and also our collagen membranes. And she showed that the scarring effect of the injury on keratocytes does not occur on our collagen. Why? Because the keratocytes are picking up a message from our collagen, align this way. Do not go fibrotic. Be normal when you come back from this trauma. And this was really our first inkling that our collagens were talking to cells and telling them to be normal. This is work from Toronto. These are cardiomyocytes, where you can see against normal collagen, ours causes the cardiomyocytes to align, as you would find in the heart muscle. And these actually will start beating with a slight challenge in the laboratory. You will get beating out of these cardiomyocytes. And this work will be published very, very shortly. This is a strip of our collagen. We dumped a group of neurons on it. Collagen is aligned this way. The crimps in the collagen are aligned up and down. That gives collagen a little flexibility. The neurites grow left to right, right to left. They know to follow the alignment. They do not follow the crimps in the material. If I was doing some spinal work or neuron work, you would want those cells to know which direction to grow in and where am I supposed to go. I mentioned before the threads and the particles that we manufacture. Here we show how we can actually populate our threads and particles with living cells. Okay, they're very happy living in our various collagens. George Martin, who used to be at the NIH, did this early work with us where he put melanoma cells in the threads. He sewed the threads into the gut of a bunch of mice, closed up the mice, came back 3 weeks later. Those melanoma cells were happy, they were proliferating, and mainly They were where he had planted them, where he had sown them. This is a problem in the stem cell world, right? You inject 4 billion cells or something, and God knows where most of them go. We can just take them and put them right where they belong, where you want them to take action. We are not a stem cell company. We are, I think you'd call us a specialty tissue engineering company. Now, John Cook at Stanford said, wow, that's really cool, Jack. So he did an experiment where he took our thread and he put microendothelial cells in the thread. And on the rats, he injected thread alone. He placed thread alone. He placed thread with cells in it, and he placed just cells. And very quickly you could see that the threads without cells, nothing happened, obviously. The threads with the cells continued to show growth and proliferation, and where he injected the cells, boom, they were gone. When he took a look, this is a Doppler